Welcome to a SNAV podcast.
Hello, everyone, and welcome to another episode of the SNEV podcast. I'm your host today, Jonas, and we have a very, very special guest with us today. Would you like to introduce yourself?
Yeah, hi Jonas. My name is Esther Nolte. Hoen. I am an associate professor at Utrecht University in the Netherlands, and I'm very happy to be here with you for this podcast.
We're very happy to have you too, Esther. It's a big privilege for us. Okay, so the first question that we always like to start with all of our guests is what sparked your initial interest in extracellular vesicles?
Yeah, that answer goes way back because it basically goes back to when I was a PhD student with Professor Marco Bouwen in Utrecht. I was working on an immunology-related topic, the crosstalk between dendritic cells and T cells and regulatory T cells. And when I looked carefully at my experiments, every time I saw some membrane proteins of the one cell type ending up on the other cell type. And I knew for sure that those membrane proteins could not be expressed by the other cell types. So this made me really think. It puzzled me. I looked back in literature. I could even find some evidence that people had seen it before, but they didn't really seem to pay a lot of attention to it. So it did kind of intrigue me, and this is why I looked a little bit further into a topic of can cells at all exchange membrane proteins via some kind of way. And then in literature I bumped into a few papers on what was called then exosomes. All the data back from 80s, 90s of the last century. So, and then at some point I thought, well, let me just look whether some of those particles are also there in my cell cultures. And well, this is how it all started.
It's always interesting to know because you started first from an immunology background, and if I remember, a lot of the early work on exosomes and EVs was to do with immunology also.
That is right, indeed. Yeah, it was really about a crosstalk between immune cells and how they could influence each other's function by exchanging these extracellular vesicles. Yeah.
Really cool. And so now you work on EVs and viruses. So could you tell us a bit more about that?
Yes. A few years back, I had to write a very big grant. I actually went for an ERC starting grant, and those topics need to be very innovative and out of the box. And there was one thing that I encountered in my research on analyzing the RNA content of extracellular vesicles. I can tell a little bit about it actually. So people had been looking at microRNAs and messenger RNAs in extracellular vesicles before I started this work. We all know the Valadi paper of the group of Jan Lutval, where indeed the focus was on the microRNA and the messenger RNA content of these extracellular vesicles. And the thing I was trying to do at that time is looking at the full range of small RNAs present in those extracellular vesicles. And so we did an unbiased small RNA-seq analysis, and I encountered many different RNA types that I had not even sometimes heard of myself before. And one of those RNA types was actually the yRNA. And I had never heard of yRNA before, so I dove into the literature, and there was not that much there around. But then I encountered some articles on retroviruses that also seemed to take those host RNAs into their virus particle quite selectively. So, and then that triggered a whole range of thoughts, I would say, because, I mean, there had already been some old papers that there might be a relationship between extracellular vesicles and viruses, because they're a little bit lookalikes. Sometimes they use the same pathways for formation from the cell, and apparently they could also share some components. And then I thought, well, there is something to learn about viruses for our extracellular vesicle field. For example, what this kind of RNAs could have as a function, because if it had a function in a virus particle, perhaps it could have a similar function when it was taken into an extracellular vesicle as well. So that is how I started thinking about the relationship between extracellular vesicles and viruses. And then I thought, okay, how to work on this topic? And there I saw some hurdles, because the enveloped viruses and extracellular vesicles are extremely much alike in terms of biophysical properties. So I thought, okay, this is going to be way too difficult to start on. Perhaps I should start on a different type of viruses first, and see whether these also interfere in the whole extracellular vesicle release pathway. And then I went to my colleagues on the virology department. They were working on picornaviruses. They are quite simple viruses, small viruses encoding only about 10 or 11 proteins. And this is actually how we started our research line on looking whether those kind of viruses were actually influencing the type of EVs that were released by cells.
Yeah, and these EVs that are packaging these non-enveloped viruses, I'm guessing they contribute both to viral transmission, but also antiviral responses from host cells too.
Yes, that could be, but I think at this point we dare still have many more questions and answers, because at first sight you would think this would be really a pro-viral mechanism, because these picornaviruses, and we work on two of them mainly, the Coxsackie virus B3 and the encephalomyocarditis virus. They early in infection get packaged inside these extracellular vesicles, and they don't encode any membrane protein. So everything in this EV membrane is host derived. So for the immune system, this is really a cover-up, basically. So nothing seems like there is something in the EV that looks like a virus. So it is also really for those viruses a way to escape the action of neutralizing antibodies. And in that regard, because they can then infect new cells and they are not seen by neutralizing antibodies, it would seem especially a pro-viral mechanism. But we were wondering whether that arm would be our only rescue here, or whether perhaps other arms of the immune system would be able to handle this kind of EV enclosed viruses. So more recently, some PhD students in my group, they started to work on is there something to recognize in these EV enclosed viruses by other types of immune cells, those involved in the innate immune system. And we recently obtained a nice data set showing that indeed monocytes can bind, take up, and also respond to these EV enclosed viruses by producing some strong antiviral cytokines. So now it seems that again, like it is with the battle between host and viruses, that there is something good in it for the virus. It can take it to spread unseen to new cells. But luckily also we seem to have developed some tools within our immune system to also recognize and respond to those EV enclosed viruses.
And with the viruses you mentioned, the Coxsackie and the encephalomyocarditis viruses, I'm guessing they have a very high tropism. They can affect many different cell types.
Yes, that's right. If there is a full-blown infection, you can see replication in multiple organs. So apparently they can infect a lot of different cell types. For Coxsackie, it is known which of the receptors actually binds and this virus, and this determines also at least part of their tropism. But yeah, I always say the EV also changes a little bit the face of the virus. So and vesicles have other receptors for binding to cells than viruses themselves. So this is something where we have very few answers yet, like whether there is other mechanisms via which EV enclosed viruses can enter cells, and perhaps use some other mechanisms to shuttle the viral RNA into the cytoplasm and start a new infection. So it might be that the EVs also contribute to making a broader tropism for this kind of viruses. But yeah, there is also some single papers that suggest that that is indeed the case. But I think there we really need to do more work. Yeah.
It sounds super interesting. And also, just to follow up, I remember the very cool review that you did with, I think it was at PNAS in 2016, about extracellular vesicles and viruses, and how closely related to are. And I think that really opened up the field and a lot of ideas for lots of people to work on this sort of field.
Yeah. When we wrote it, it was a very, very nice experience together with Leonid Margolis. Because it triggered a lot of thoughts, and eventually this was at that time more a viewpoint that you would have for the enveloped viruses a whole continuum of different particles, where at one end you would have the fully infectious viruses, and then in the middle there would be intermediate structures of non-infectious viruses, VLPs, EVs that contain both host and virus proteins, and on the extreme other ends there would be vesicles with only host proteins. And it is very interesting because at many conferences that I go to, people show this scheme of the continuum, and apparently it triggered a lot of thoughts about the relationship between viruses and vesicles, and that first of all, there's something to learn, but second of all, it is quite difficult to really disentangle who is who in this continuum, and how to kind of use proper methodology to investigate which of the particles has which function.
No, it's a really, really cool idea. And who knows, maybe in the future, ICEV will start to swell up some of the virology fields.
Well, I mean, I really would stimulate this kind of interactions more and more. I mean, I also know that within the EV field, we try to connect more and more to different disciplines and expert fields. And I think that is extremely important because, in many different research fields, the EV pop up as a potential contributor, for example, to disease or homeostatic processes. But the key will be to combine our EV expertise with the expertise of those researchers to really get to the answers for those specific fields.
That's very, very true. So going back to, I think earlier you mentioned RNA within EVs, and what do you think makes this RNA present in EVs particularly significant? Considering people often, when they work in the EV field, they tend to really focus on proteins and sometimes lipids, but very rarely do people ever look at RNA unless it's microRNA or other things. What do you think makes this RNA within EVs very significant?
That is quite a difficult question. And I think the strongest data that we have is that the RNA content of the vesicles are representing, in a way, what is going on in the cell that releases them, and then hence also the interest in the biomarker field for analyzing these EV enclosed RNAs. But I think there's two aspects which make it quite difficult to really pinpoint what the function of these RNAs in the vesicles could be. First of all, it is that there is a lot of RNA types in those vesicles, of which sometimes we know the function inside cells, but we have no idea what their function would be outside cells or when it is transferred into a new cell. That is one thing where there's still a lot of questions. And then the other difficult thing to answer is, would it eventually end up in the target cell where it can actually do its function? Because if it is in the lumen of the vesicle and you would need it in the cytoplasm of the target cell, that would mean that indeed the vesicle needs to fuse with the target cell. And I think that is where the vesicle tries to solve or get the pieces of the puzzle into the right place at the moment, because in our experimental in vitro models, this fusion seems to be quite rare, meaning that, probably the functional delivery of those RNAs into the target cell may also be rare. But maybe we don't have the right environmental conditions in our in vitro systems to make this delivery of the RNA effective. How do we know? There are some in vivo data where indeed there is evidence that EV RNAs can be functionally delivered to target cells in specific tissues. So maybe in vivo this process is even more effective than what we try to mimic in our in vitro systems. But this makes also the studying of exactly the RNA content and its function in target cells a very cumbersome process, where I think we need better tools or better conditions to be able to study this properly.
And also the role of RNA binding proteins must be also important. I'm thinking back to the recent work of you and Tom, who we very much know in the SNEV community, in the community.
Yes, I think for specific shuttling into extracellular vesicles, something specific must also happen. Either localization into a specific area is very important, or perhaps the shuttling with the help of RNA binding process into vesicles is particularly important. And we know for some of the RNAs what are the RNA binding proteins, and this is also the study of Tom Driedongs within my group. Like, can we now follow within the cell where those specific RNAs and their RNA binding proteins are during the vesicle release, and can we follow that on their way to the outside of the cells? And it is never so that one RNA has only one RNA binding protein. That's quite rare. So first of all, the idea is really to find out which of the RNA binding proteins is then specifically involved in the routing of the RNA into the vesicles. And then the question is also, is it then still functional in the extracellular vesicle, or is it just bringing the RNA to the right spot but then doesn't go into the extracellular vesicle together with the RNA? So I think we only got a little tip of the iceberg there in that paper, but it should also stimulate thoughts on how to exactly address these kind of questions.
Definitely, indeed. And also, I think the tools and the mechanisms and the machines that we use to try and isolate these different EV subtypes are important. And that leads us to our next question. I remember correctly that you were one of the first people to kind of do flow cytometry on EVs. And I think a while back we invited Estefania, who gave an amazing talk in the SNEV talk series. I think a lot of people really enjoyed her talk, and it's still available to listen to now on YouTube. What do you believe has been the biggest challenge in trying to use this method to study EVs, and how do you feel as if the flow cytometry has really helped to push EV characterization?
Yeah, when we started that technique, that is really way back, together with a very skilled operator, the operator before Esther, basically, in our facility, who was very much intrigued and interested in seeing how much we can actually push existing flow cytometers to detecting very, very small particles. And I think the biggest challenge that we had was not so much getting the dots into your screen, because flow cytometers always detect something. No, I mean, that is not the challenge. But the real challenge was, is the dot that you see on the screen really an EV? Is it an EV subtype? Is it the background, the noise? So trying to figure out what the machine actually saw, and how it translated that to a signal in our detectors. That was one challenge, but the other challenge was also how to process your sample, labels your sample so effectively that the chance of getting it in the right signal to noise ratio in the machine. Yeah, that was one of the biggest challenges that we had. So it was working on the labeling of vesicles on the one hand, working on reducing the noise and reducing contaminants on the other hand, and on the third hand, which you don't have, but on the third hand, you also need to work on the sensitivity of the machine. And this is how we encountered all kind of issues. For example, if your concentration of particles too high, you see a lot of events, but actually it's a sum up of multiple particles. But this is something you will only see when you start to dilute your sample, and suddenly you see with the dilution that the numbers increase instead of decrease. So every time that we measured something, we had a surprise that we couldn't really understand based on what we knew. And every time we had to do multiple more experiments to figure out what explained actually those results. So this is why setting up that technique really cost a lot of time. But then I think because it was one of the first techniques that could really look at single vesicles based on multiple parameters, yeah, that was something that did not exist at that time. And nowadays we have many more techniques that can do the same. I think the advantage still of flow cytometry is the speed at which you can detect and quantify the vesicle subsets, because if you can analyze 3,000 or 4,000 events per second on multiple parameters, I think that is still a big advantage of flow cytometry.
Yeah, and I think there's now more and more people using flow cytometry to characterize the EVs. If I remember, there's a whole framework that's being used, the MyFlowSite EV, which you spoke to last time with Joshua, who gave us a very nice description of that. Do you still see that being a very, very important framework that should still be used and as a guideline to help people use flow cytometry in their EV experiments in the future?
As the biggest advantage, I see that people should be aware of many different things before they start working with this technique. That is one big advantage. So because it is so easy to turn on a machine, run a sample, and just record what you get, I mean, that is very easy. But knowing what that means, that is really something that you can only do when you have gone through all the different controls and steps that are described in that framework. And so this point of realization is very important. And then the second important aspect is that we really need to report well, because otherwise it will be impossible to compare our data. And that is also something that is described elaborately in this framework. And like many other aspects of the EV research, EV track tries to aim for the same. Reporting is hugely important for any technique, including the flow cytometry.
Yeah. So yeah, definitely. I think reproducibility is also, as you mentioned, is very, very important. Just so that we all are on the same page.
Yeah.
Especially as you mentioned, flow cytometry is can easily be misleading.
Exactly. That's exactly what I mean. Yeah.
So I think just earlier this week, you were at the Netherlands Society of Extracellular Vesicles annual meeting, and I'm sure you saw lots of amazing talks on all the amazing work being done in the Netherlands in the field of extracellular vesicles. And you were a co-founder of that society, and we wanted to know, how did you go about setting up this society, and maybe you could give some ideas for people who are in other nations where the EV field is just starting to grow and maybe they want to start their own society.
Yeah, the first step is getting together with a group of EV researchers within your country that are really enthusiastic about the field and see the importance of bringing a community together within the country that you live in. And we saw this importance because at that time there were not that many EV researchers yet, so especially within one country. We thought it is so much better to get to know each other, to share knowledge, to share equipment if needed. And that was really the main reason to start up the society. And the way we did it was that we grouped a couple of researchers that we knew already from the field and who had the same enthusiasm as we had. And then we just started it. And I think the advice is you can start small. That doesn't matter. Your community can start small. It will grow eventually and sometimes even quite fast, especially if there's a lot of groups that already work on the topic within your country.
And within the Netherlands, I mean, now there's so many leading labs from the Netherlands who do a lot of very pioneering work on EVs. How has collaboration been over there? I mean, I'm guessing it's been fantastic considering all the publications that's been coming out.
Yeah, quite funny. From the beginning, we had actually the idea that that people had already quite some collaborations abroad, but not so much within the country. And I think that's just historically how things grew. And sometimes you don't even know that people also work on EV, even though they're in a city like only 30 kilometers away from you. And when we started to make known that we are building a society and that we were asking people to join and to share what they were working on, and that then things started to move a little bit faster, and we were indeed sharing reagents, letting each other know where the equipment was, and that's actually how it went. And currently, big grants are being written also together, and there's a lot of interactions between EV researchers in the Netherlands.
So the next question I wanted to ask is, you've done a lot of work on translational EV research, as you worked with EVs, with viruses, and on other topics too. Where do you see the progress has been made in translating this EV knowledge that we have into diagnostic tools and treatment?
Yeah, I'm very much in favor of also not only translating, but also working on mechanistic parts of the extracellular vesicle communication. Because I often think that translation is in need of more mechanistic work, because we then can better know how to use the extracellular vesicle for diagnostic purposes, but especially also for therapeutic purposes. So I think both the translational and the more fundamental science should really go hand in hand. And the translation is very important to really see where the field can be heading and to keep the interest in the field. And some parts of the fields are already way more developed. For example, the mesenchymal stromal cell EV applications, they are quite advanced already. But for other types of vesicles, I think we also still need a lot more fundamental knowledge to be able to see is there any kind of cell type, target cell type specificity of those vesicles? What are their signaling properties? Are those coming really from the outside membrane of the vesicles? Is it the luminal content that is really important? And also, how can we manipulate those vesicles to do an even better job in the therapeutic applications that we think they might be useful for? And in this regard, I think it is also interesting to not only look at the mammalian field, but broaden our view also to other kingdoms of life. And there are initiatives already that also plant EVs are studied as potential therapeutic entities. But this can be broadened out a lot more. So within the coast action, so I'm in a EU coast action, there we try to bring together people from EV fields across all kingdoms of life. So people that work on plant EV, on fungi, on bacterial EVs, and there is really so much more to learn about how other organisms exchange these EVs and use them for communication properties. That perhaps some of those properties we can also translate to our own engineered EVs to make them even more fit for whatever purpose we want to use them for.
So yeah, focusing on the mechanistics then in that sense. One of the key questions in the field is always trying to look at the site of origin of EVs, whether or not these EVs are derived directly from the plasma membrane, whether they come from an endosomal pathway, or maybe if they bud directly off a particular organelles. Is this where you see where the most important part of EV biology is going to come from in the next couple of years, where people need to really decipher these kind of pathways?
I definitely think that we need more knowledge there, but we've also learned in the recent years that those pathways are extremely complex. We have different pathways for EV biogenesis, but they're not strict pathways. There's a lot of crosstalk also between those pathways. We are, for example, also working on the interplay between the autophagy pathway and extracellular vesicle release. And that is very important because whatever pathway the vesicle will take to come to the outside world, it will also determine what kind of composition these vesicles have. So either for understanding the EV communication, for example, in disease processes, or being able to engineer the extracellular vesicle so that it has the properties that we want, I think knowing more about how they're formed and how they're released is extremely important. Equally important as to where they end up when they are released, whether they can travel a lot of distance or whether they get stuck at the ECM, the extracellular matrix, and when they reach a target cell, what is then still needed for them to actually exert a biological effect. So I think at all these three stages, we really need to build up more knowledge to be able to translate it even better for therapeutic or diagnostic applications.
I definitely agree. And then we'll start with our final question, which we always like to ask to our guests, is if we were to go back to your work 10 years from now, and we go 10 years into the future, where do you hope will be the major breakthroughs in EV research, and how do you think they'll impact our current ideas?
I think the breakthroughs will need to come from what kind of conditions are really crucial for vesicles to be either used as communication entities or perhaps for their purpose to get rid of certain components from cells. And this is something, it is something big, so that is not something small, but I think in 10 years we will need to have a better idea about that, to really look more into the physiological function of EVs. Because we know that for almost all biological processes, EVs are part of, for example, the cellular or the inter-organ communication pathways. And what we're trying to do now is isolate the vesicles and then see what they do in a certain system, label them and see where they end up, and then perhaps characterize the cell where it ends up and whether it's really changed or not. But I think our challenge in the coming years also lies in how do these EVs work together with all the other communication systems that cells and organs have. And that comes down to doing also proper in vivo experiments and trying to really unravel the contribution of the EV in the total of the communication between organs and cells. And that will maybe not so much tell us more about therapeutic applications or help us in diagnostics, but it will really help us understand the physiological and the pathological role of EVs in the complete system.
No, I definitely agree. Even the idea of isolating specific subtypes of EVs, and especially what we do a lot in vitro in cells, is we tend to pick one small group of these EVs and study their physiological role in whether it's cell signaling or delivery or whatever. And sometimes by isolating this one thing, we kind of miss, how it's involved with the rest of the EVs there too. Maybe they also play a part, and we don't know exactly.
Yeah, I totally agree. And it is a way, we need to simplify also to answer questions. I think that would be the first step, to really isolate the population and see what that population does. Then perhaps also see what the other population does, but in the end you will also need to see what the whole picture is and put the population together and see what the population has in its fullness for effect on cells, and perhaps then also with other components that are at the outside milieu of cells.
Definitely. Well, just want to say thank you very much for your time, Esther. It's been great talking to you. I really appreciate you giving us your time, and also say thank you for all the amazing work you've been doing with EVs and viruses and flow cytometry also. I think it's been very encouraging for young researchers. Is there anything you'd like to say to SNEV listeners in particular? Anything for them to look out for?
I would say keep up the good work. This field really has so many open questions, and it might sometimes be a bit tough to get to the right technologies, to ask the right questions. But this is a fun field to work in because there are so many things that are still there to discover. And keep up the good work with SNEV, because I really, really appreciate the initiative. And I think by connecting the early career researchers, this is such an advantage for each of you also to discuss and to come up with new viewpoints, to share your knowledge. So good luck with that in the near future.
Thank you very much, Esther.
Thank you.